Let's continue our discussion of energy and talk about enzymes. So we said that all of the metabolic pathways in a cell are mediated by enzymes. This means that enzymes are controlling when, how, and how quickly a reaction takes place. This is critical in a cell because you need to moderate the reactions based on the needs of the cell. If all reactions went whenever they needed, we would probably be consumed by heat and just burst into flames. That might be a bit extreme, but it's a good illustration about showing how enzymes actually work in a cell to control the progression of these pathways. Remember that we talked about ATP, our energy currency in the cell, and that in the process of providing energy to a chemical reaction, ATP is going to transfer a phosphate group to that reaction, and our energy reactions are coupled, meaning that exergonic reactions power endergonic reactions. So this is all related to enzymes because enzymes mediate all of these processes. They mediate the transfer of phosphates, they mediate the actual reactions that occur. So enzymes are important because they speed up the metabolic reactions in a living organism. A lot of our reactions are endergonic, they are non-spontaneous, which means they are not going to happen on their own. Or they are exergonic reactions that are going to release energy, but require a little bit of a nudge to get them going. So enzymes are proteins that are acting as a catalyst. So a catalyst is a chemical agent that speeds up a chemical reaction. In this case, it happens to be a biological molecule, a protein. Enzymes and catalysts are not consumed in the reaction. So they are used to speed up the reaction, but they are not consumed, which means they can be used over and over again. Anytime you see ASE at the end of a word in biology, it probably means that it is an enzyme. It is just a ending to the word that indicates that protein is acting as an enzyme. So in this reaction we have sucrose. We have a glucose and a fructose bound together by a glycosidic linkage. And let's say that the cell wants to break this down. It wants to use the glucose or the fructose. This is a dehydration reaction. We've seen this before, but what we haven't discussed is the fact that that dehydration reaction is actually controlled by an enzyme. In this case, it is the enzyme sucrase. Enzymes are specific in binding only specific reactants and changing them in a way to produce products. So in this case, our sucrase enzyme is going to specifically bind that sucrose molecule and break that glycosidic linkage. So it's going to help perform that hydrolysis reaction. Enzymes work on what we call the activation energy barrier. In a chemical reaction, they proceed toward the products when the products and the reaction when the reactants interact. They react because they are more unstable separately than they are together, or they are more unstable together than they are separately. And so in a chemical reaction, you need to take unstable molecules and put them into a more stable product. Energy has to be put into the bonds to change them. Even if it is an exergonic reaction, you still need a little bit of energy to get the reaction to go. But ultimately, you are releasing or putting in energy that is going to make more stable products. So enzymes work actually, let me back up a little bit. Activation energy can be thought of as sort of a little bit of energy investment. It's just a little bit or maybe a lot of energy that's needed to start a reaction. This energy is needed even in an exergonic one where energy input, normally you have energy put out, but you need a little bit of energy to sort of make those bonds unstable enough to go to their products. So our graph here shows our reactants, A to B and C to D, two imaginary molecules. That energy of activation, that energy that is used in this reaction, is going to cause those bonds to be unstable. And then because they are unstable, they're going to react so that they can produce a more stable form. In this case, we have A bound to C and B bound to D in our products. This is an exergonic reaction overall. There's a net release in energy, even though we needed to put a little bit of energy in to get it to go. The activation energy can come from one of two places. It can either come from enzymes, or it can, well, sort of, or it can come from heat in the surroundings. So very often, activation energy is simply provided by the heat in the surroundings, the heat around the reaction. So our bodies produce heat, and sometimes this is enough to drive that activation energy and to drive the reaction to completion. But the problem is that some reactions have such a high activation energy that heat isn't enough, or the heat is too much for the organism to handle. I know this says discuss with your neighbor. I would like you to think about why heat is a problem, why organisms can't just continue to heat up to overcome the activation energy required for getting their reactions to go. So heat is a problem because it can cause damage to an organism. And there's a maximum heat that organisms can withstand, either internally or from their environment. And so while heat can provide a little bit of energy, it can't always provide all of the activation energy needed for a chemical reaction. The other problem is that heat speeds up all chemical reactions. It can put in energy for non-specifically for activation energy. And so you get to a point where if you put in too much heat, you're going to be causing all the reactions to go, not just the one that you're needing. So we need something a little bit more specific to provide the activation energy for specific reactions. This is where enzymes come in. A second ago, I misspoke and said that enzymes provide activation energy. What I actually mean is that enzymes lower activation energy. They don't actually add any energy to the system. Heat adds energy to the system. Enzymes instead affect the reactants in a way that they don't need as much energy to become unstable and for the reaction to proceed. So enzymes lower the amount of activation energy required. This means that the enzymes can actually bring the reactants to an activation energy that's low enough to be overcome by the heat in the environment. So enzymes can change the rate of the reaction, meaning that they change how quickly they will get started, but they don't change the energy actually input or the actual energy released. So this graph is key to understanding what's going on with activation energy. The black line shows the energy required in a reaction without an enzyme. So we have our products that have a high amount of energy, our reactants have a high amount of energy compared to our products. This means that we are going to have a negative delta G, or a net release in energy. The red line shows what happens when you add an enzyme to the system. So the red line is showing that an enzyme has been added to the situation and it has lowered the activation energy. This just means that there's less energy required to get it started. It does not mean that it has changed the total amount of energy released from the system. Because if you look, our reactants have the same amount of energy, whether they are being mediated by enzymes or not. And we release the same amount of energy, whether or not there's an enzyme present. The enzyme just affects how quickly that reaction will start. So let's talk about how enzymes actually do this. And I need to go through a few vocabulary words first. The first one is the substrate. The substrate is just the reactant that the enzyme's going to work on. Enzymes are extremely specific. They usually work on one reactant and one reactant only. And this allows them to be very specific within the cell, to perform very specific duties within the cell. Remember that an enzyme is a protein, and a protein is very dependent on its structure for proper function. Part of the structure of an enzyme is the active site. So the active site is the region where the substrate is going to bind to the enzyme. So we have this blue enzyme molecule here, and we have a little red substrate. That substrate is going to bind very specifically to that active site. Once that substrate binds, the enzyme will actually change shape a little bit and sort of tighten and hold that substrate in. This is called induced fit, and it is key to the function of the protein. So, how do enzymes actually lower the activation energy? Well, it can happen in a few different ways. One is that the active site simply brings the reactants close together so that they are in close proximity and have a better chance of reacting. The other is that it can stretch one or more of the molecules to make them unstable. And remember, when they're unstable, they're more likely to react. They can also provide a specific microenvironment for the reaction. So let's say your reaction requires an acidic environment, but your cell environment's not particularly acidic. Based on the shape of the protein, that active site might actually form an acidic environment for that reaction, which would allow it to go. And finally, the enzyme might actually covalently bond to the reaction. This is going to cause a chemical change in the reactant that is part of its conversion to a product. This diagram here shows how an enzyme might bring two substrates together. So starting at the top there, we have, in this case, two substrates, so two reactants. They're both going to bind specifically to the active site and form what we call an enzyme substrate complex. This is just all of the components of the reaction plus the enzyme bound in a specific way. So here we have brought both of the reactants together, possibly produced a microenvironment that's more conducive. But then from there, those substrates that are in close proximity are going to react and be turned into products. So our products are shown in 3 there, those blue and yellow molecules. Those are our products. They're then released. As I said earlier, enzymes are not consumed in the reaction. So this enzyme can go on and bind more substrates to produce more reactions. Enzymes are often have the ability to do both the forward and the reverse reactions to mediate those. And so we could have a situation where the cell needs the products back, and so the reactants back, and so we can actually bind the products instead and turn them back into reactants, depending on what the cell needs. Remember that enzymes are proteins. And with proteins, their function is incredibly dependent on their structure. So we now need to talk about ways that enzymes might be affected. So keeping in mind that enzymes are proteins and the structure is closely tied to their function, think about some things that might cause a change in protein shape. Basically, anything that can interrupt protein structure is going to affect protein function. And this goes back to our idea of structure and function. So think about things like heat, functional groups, pH, things like that. All of those are going to interfere or potentially interfere with the enzyme's ability to function. One of the most important effects on enzyme activity is concentration. In general, in a chemical reaction, if you have a higher concentration of your reactants, the reaction is going to go faster because they're more likely to find each other to interact. And so you can increase the enzyme. This means that any substrate will be found more quickly because there are more enzymes. You can also alternately increase the substrate concentration. If you increase the substrate concentration, you're going to increase the likelihood that an enzyme is going to find its substrate. However, you can't necessarily increase either of these forever. At some point, your enzymes are going to outweigh your substrates, and you're going to sort of limit out on the number of reactions you can perform. And alternatively, you can reach something called enzyme saturation. You can add so much substrate, so many reactants to that system that the enzyme becomes saturated. That means that at any one point, all enzyme molecules are tied up with the reactant. At that point, the reaction can only go as fast as that enzyme can work, can only go as fast as it can bind the substrate, convert it into products, release it, and bind another one. Temperature is also really important for enzyme activity. If you increase the temperature surrounding a protein, you're very likely to begin to disrupt hydrogen bonds. Remember, hydrogen bonds are really important in the secondary structure of proteins, holding together the alpha helix and beta pleated sheets, which is sort of their first level of 3D structure after the polypeptide. All proteins are going to work best in a specific temperature range. That means they have a specific temperature in which they can work the fastest, the conditions are the most optimal. If you increase the temperature, you are also likely to increase enzyme activity because all of the molecules, the substrates, and the enzymes are going to be moving faster. It's more likely that they're going to come in contact. Likewise, you can decrease the reaction rate by decreasing the temperature and slowing down the molecules, slowing down the chances that they're going to collide with one another. Going back to heating a system up, if you continue to heat up the temperature, there's a point at which you're actually going to decrease your reaction rate. You're going to get the system so hot that your proteins begin to denature. They begin to unravel and become dysfunctional. So temperature works within a range. There's an optimal range, and increasing slightly, still within that range, you're going to increase your enzyme's ability to work. But too hot, and you're going to denature them. That means you're going to break the proteins apart, and they're going to become dysfunctional. pH works in a manner similar to temperature in that most enzymes are active at a specific pH range. In general, the temperature and pH range are those of the body of the living organism. So for us, the temperature, the optimal temperature is going to be around 98.6. That's our normal body temperature. And most living organisms are going to have a normal body temperature. For pH, it's the same thing. Most enzymes are going to be active around a neutral pH. When we talked about pH, we said that most enzymes are, neutral pH is between 6 and 8. So most enzymes are going to be most active around that point. As you get closer to their optimal pH range, those enzymes are going to become more active. As you move away, as you become more acidic or more basic, those enzymes again can begin to denature, just like with temperature. And that's because you have hydrogen or hydroxide ions that begin to interrupt the protein structure, begin to make that enzyme less functional. Next, we can have the requirement of something called a cofactor. So a cofactor is sort of a protein helper. It's another molecule that binds the enzyme to make it functional. Sometimes it is fully required for that enzyme to function. Other times it just helps it to be more effective. These are often inorganic atoms, organic coenzymes. These are organic molecules that bind the enzymes. And what they do is when they bind, they cause a shape change that makes that protein better able to bind its substrate. We can also inhibit our enzyme activity by adding certain chemicals. So depending on the enzyme, you may be able to add what we call an inhibitor. There are two types of inhibitors. Competitive inhibitors bind at the substrate. So they block the active site so that your substrate cannot bind. So they are going to be somehow similarly shaped to your substrate, and they block the enzyme from functioning. They cause a shape change when they're bound and keep your enzyme from working on any of the substrates. A non-competitive inhibitor binds a location elsewhere on the enzyme. So it doesn't bind at the active site. It doesn't block the active site. But when it does bind to that alternate location, it causes a shape change in the protein to make it dysfunctional. Both of these situations are probably going to be temporary as ways to block a reaction from going forward. Finally, one of the ways that a cell regulates the activity of an enzyme is by something called feedback inhibition. And it uses the idea of either a competitive or a non-competitive inhibitor in order to block some point in a metabolic pathway. So up here we have the conversion of threonine into isoleucine, which is an amino acid, by an enzyme, or actually by a series of enzymes to mediate that metabolic pathway. In feedback inhibition, the end product actually binds one or more of the enzymes in the pathway and blocks that pathway from happening. So as isoleucine accumulates, as the cell begins to have enough isoleucine, it's going to start to compete with the threonine, and it's going to bind to the active site and block that reaction from happening. This is one way that cells control the products in their cell. You don't want to have an accumulation of products forever. These metabolic pathways require energy, and so you only want to produce what you need. You also don't want to store things that you don't need at the moment. So feedback inhibition is one of the mechanisms that cells use to control what is actually being produced in their cells.